Hello, my project is called Protein Misfolding Patterns in Rare Disease Linked Proteins using AlphaFold 2. I'm studying whether protein structure can explain why some rare diseases become much more severe than others. But first, a little bit of background. We'll start with the basics. Genes. Genes contain instructions for making proteins. Proteins do almost everything inside of a cell, but only if they can fold into the right 3D shape. Mutation changes a protein sequence, it can change how a protein folds. And you can see, for example, here on the right side of the screen, you can see the difference between a healthy protein and a protein that is folded wrong. When it folds wrong, it may stop working, get destroyed, or form toxic clumps. This is what we call protein misfolding. Rare diseases together affect about 6% of the world, and many of them are caused by misfolded proteins. You can see we have some examples of diseases, and genes like CFTR, when they make misfolded proteins, can lead to diseases like cystic fibrosis and other examples shown. The problem is that we don't often know how severe a disease will be until the damage is already happening. So being able to predict severity early really matters. Enter AlphaFold. It is an AI system developed by Google DeepMind that predicts a protein's 3D structure from its sequence and allows us to get a confidence score called PLDDT. A high PLDDT means you have a stable structure, while a low PLDDT, especially below 50, usually means instability or disorder. Now, we get to here. My research question, put simply, is do proteins linked to more severe rare diseases show lower AlphaFold confidence than proteins linked to milder diseases? The purpose overall was to test whether computational programs like AlphaFold can help to estimate disease severity early on. And my independent variables were the genes being studied and the severity across variants, while the dependent variables were the actual mean PLDDT and fraction of the residues below 50. My hypothesis was that proteins linked to more severe diseases would have lower PLDDT scores and more low confidence reasons. And I used ClinVar to find the actual pathogenic variants, OMIM, to get actual markings for disease severity, and UniProt for protein sequences. I collected about 13 proteins and split them equally into mild, moderate, and severe groups. Now, onto the actual procedures. I first, after getting ClinVar for the variants and OMIM to classify diseases into mild, moderate, and severe, downloaded protein structures from UniProt and ran them through AlphaFold using CollabFold. AlphaFold produced a structure and per residue PLDDT score for each gene and protein, and then I wrote a Python script to calculate the mean PLDDT, fraction below 50, and variability. And I visualized these structures using PyMol using blue for high confidence and red for low confidence, and ran statistical tests comparing severity groups. Now, we can look at a structural example here of what the visualization actually looks like, where blue regions mean stable folding, and red regions or other colored regions are unstable or disordered regions that are likely to actually interfere with protein function. Now, the actual results are showing us how many of the severe proteins have more of these colored sections or more of these unstable regions? And you can see in the scatter plot, overall, as disease severity increased, predicted protein confidence decreased, while mild protein structured at high PLDDT clustered at IPL-DDT, while severe proteins had much lower confidence, meaning the more severe disease was in general, is the less confident we were in its structure, i.e., the more likely it was to destabilize, the more severe the disease was. And you can see more statistics that show that PL-DDT and severity were strongly and significantly correlated, and mild and severe groups were clearly different. You can even see the difference in percentage of low confidence scores here. Now, overall, this supports my hypothesis. Many severe diseases involve unstable, misfolded proteins, but not all severe diseases come from misfolding, and that is a limitation that AlphaFold cannot detect every type of failure. Overall, though, this shows that AlphaFold based modeling can be a very powerful early tool for predicting disease severity and letting us know what treatments to use for what people at the right time.